Hello. Today we are going to be talking about the partial differential equations that model cancer tumor cell growth. My name is Lawson, and my partner for this project was Ximing Yao, and our mentor was Dr. Olga Turanova. To begin with, we are going to go through an introduction that allows us to go over the conceptualization of the problem and how exactly we frame our tumor and its growth. To begin with, our model considers the tumor to be some subset of Euclidean space, which we call omega of t. It's kind of shown over here to the right as a two-dimensional blob. Further, we need to consider how exactly we will talk about the change in the tumor. To do this, we first need to talk about the boundary of the tumor. At every point along the boundary, we will define an outward pointing normal, which we will call eta of x and t. Further, we need to find some way to talk about the change in this boundary. And to do so, we will talk about its velocity in the direction of each and every one of these outward pointing normals. To define the velocity, we are going to define a helper function w, which its gradient will help us give the velocity of the boundary at any different direction of the outward pointing normals. Now, because the velocity describes the change in the boundary of the region, it describes the change in the region itself. However, this function w is dependent upon a system of partial differential equations, which are determined by that region itself. And so then that will help us define a new velocity, which will help us find a new region and a new velocity back and forth in this sort of cyclical relationship. Next, we'll talk about the different types of models. For our type of model, we are using an incompressible type, which helps us give a relationship between the density of the cells, here called N, and the pressure of the cells, here called P. The relationship between the two is called the constitutive law for the model. For an incompressible type, all we have is that the density of the cells is 1 anywhere the pressure is greater than 0, and is 0 anywhere the pressure is 0 or negative. Now we can talk about the generalized n-dimensional model, which is shown here as equation 2. This first equation on the left-hand side, we have the description of the motion of our cells. And on the right-hand side, we have the limits upon the growth of the cells itself. And then our second equation gives us the form that allows us to talk about the motion, which is determined by physical principles and fluid mechanics. It's useful to look at this as we will move on to a more specific model in one dimension. In one dimension, we have a whole bunch of very useful properties and factors which allow us to simplify down that model and find a very explicit formulation of our problem, which allows us to study a whole bunch of very interesting properties. So in one dimension, our region omega of t may be considered to be symmetric about the origin and defined off of some function r of t, where r is a function from the positive reals into the positive reals. Further, in one dimension, we only have two outward pointing normals, those being eta of plus or minus r, which are exactly plus or minus 1. And further, we only have one spatial dimension to consider, and so our gradient of w reduces simply down to the partial derivative of w with respect to x. Now that we have all of those considerations out of the way, we can work on changing our model from that n-dimensional type down to the one-dimensional type. So the pressure in one dimension is considered as a function of both w and a new consideration, which we call nu, which is the friction between our cells and is some value between 0 and 1. This function is shown here in equation 3, and we can see that it is some value on our region omega of t and is simply 0 everywhere outside. So referring back to our constitutive law, we can see that anywhere in our tumor, we have a density of 1, and anywhere outside of our tumor, we would have a density of 0. Further, it should be noted that our function for pressure will not in general be continuous, and we would expect some sort of discontinuity at the boundary r of t and negative r of t. Using this expression for p, we can now talk about our model for incompressible growth, which is shown here in equation 4. Which again is just a simplified version of that n-dimensional model. This first one is the one that helps us describe the motion itself and allows us to solve for very explicit expression for w. And the second one describes the change in the boundary of our tumor cell. And so that will allow us, once we have an expression for w, to try and determine what exactly happens to our tumor with time. So now that we have our model derived, we can look at our goals for our research. Our goals are twofold. Firstly, we want to be able to describe the behavior of a tumor for some large time t. And secondly, we want to identify the role that friction plays in the model. In order to do so, we will first explicitly solve for w of xt for some fixed time t. This will allow us to work with the model and determine what happens to the tumor's boundary in time. Next, in order to identify the role of friction, our goal will be to, well, remove friction from the system itself by taking a limit as friction goes to 0.
Well, as Riley talked about how we build the model, then I will show you how to solve it. Substitute the equation of pressure into the model, we get the system as follows. Observing these two formulas, we find that this is an ODE problem. Using the relevant knowledge of ODE eigenvalues, we can get the expression of W. We number the solutions of these two equations. W1 describes the state of the model on R minus interval minus RT, and W2 characterizes the model on the interval minus RT, where the coefficients of C1, C2, C3, and C4 are all unknown. Next, we focus on the determination of these coefficients. We want to smooth the function, which means the equations of W1 and W2 are expected to be continuous at RT, so we can write the formulas like the slides. In our hypothesis, W is symmetric in the two intervals minus RT0 and 0 RT, which infers the derivative value of W as 0 is 0. Besides, we think W is smaller and smaller as it keeps away the tumor, considered with the expression of W1, which we can conclude that C1 equals 0. The specific coefficient expression form is shown in the slides, which looks a bit complicated. To make the results clearer, we did a numerical simulation. In this special case, we set nu equals 1 and t equals 1, t equals 5 like this. The blue line in the picture is W1 and the red one is W2. We can observe how W changes over time, and it perfectly meets our assumptions before. In other words, W is what we're looking for. Then we calculate RT by the following equation. Note that the expression of C2 as we showed before contains RT, and the expression of R prime also contains C2. This is a closed loop system, and it is very difficult to solve, so we estimate the upper and the lower bounds of RT, and the result is shown in the figure below. It's surprising that the tumor's growth is linear. Now that we have all the parameters in the model, we next explore how the system will change when the friction disappears. In our model, the disappearance of the friction is equivalent to nu equals 0. We revisit the model of incompressible growth. Through the second formula about the pressure, we can get the conclusion of W. W equals P for the interval minus RT, and W equals 0 for the interval R minus the interval minus RT. Through some calculations, we can get the model without friction as follows. Then we explore some properties of W and P and got the conjecture as follows. There exists R bar, P bar satisfying the system 7 as we talked before, such that R nu converges to R bar, P nu converges to P bar, and then W nu converges to P bar uniformly as nu went to 0. This is the result we expect, and it is very useful in the model simplification procedure. That's all. Thank you.